Chapter 9, Control of Microbial Growth in the Environment. In this chapter, we're going to be covering the following topics: basic principles of microbial control, selection of microbial control agents, physical methods of microbial control, and chemical methods of microbial control. Before I move on from this overview, I want to let you know that I consider this to be one of the most important chapters in the textbook, because you're going to be using this no matter where you go, what profession you go. You can be using this information in your home. So I'm going to be testing you more on this one. Hint, 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 hint. So before we launch into this chapter, let's talk about what in the world we're talking about. Control of microbial growth. Now, in the previous chapters, we've talked about how to try to get microbes to grow in the lab. Well, do we want microbes growing outside of the lab? That is a question to consider as we go through this chapter. And we'll be doing kind of a summary at the end of the last chapter. So, when is an example of when you want to control microbial growth? Well, here's a lovely example. Beautiful strawberries that I've just picked from my garden. I've gotten a whole container full of them. Of course, I ate some while I was picking them. And 2 days later, after sitting on my counter, this is what happened. I would have liked to have controlled the growth of the fungi. Okay? So instead of leaving them on the counter, what could I have done to have controlled the growth of microbes? Now, I bring up the food example because traditionally, this is the main thing that humans have been concerned about in control of microbial growth. How do I keep my food from going bad? It's only after Pasteur and Koch and researchers during the 1800s that we became concerned about controlling microbial growth in preventing disease. These are the two things that we're going to be looking at mainly. Controlling microbial growth in food applications, health applications, and there are some industrial applications. So let's move on to our first topic, basic principles of microbial control. The learning objectives for this topic are contrast sterilization, disinfection, and asepsis, and describe their practical uses. Contrast the terms degerming, sanitization, and pasteurization. Compare the effects of static versus sidal control agents on microbial growth. Define microbial death rate and describe its significance in microbial control. Describe how antimicrobial agents act against cell walls, cytoplasmic membranes, proteins, and nucleic acids. The overview for this topic is we're going to be covering the terminology of microbial control, microbial death rates, and the action of microbial agents. First, let's talk about the terminology of antimicrobial control. You will need to learn all of the terms in this table from the book. It's a very good table. But let's go over some of the terms in more detail. First, let's start with side or sidal and stasis and static. These suffixes, when they're added to a microbial control method, tell you what they do. So if it's side or sidal, it indicates destruction or kill. Think homicidal. So for stasis and static, that inhibits growth, that inhibits metabolism, but it doesn't necessarily mean kill. So if you've put something in stasis, it's asleep. It's not necessarily dead. But that can be useful. Even though you would think that sidal would be the only one that would want to use all the time, some organisms we just can't kill. Or it's very hard to kill, or it's impractical to kill. So, if you put them into stasis, hopefully your immune system can take care of them. Now, here's another one. Degerming. This is the removal of microbes by mechanical means. A lot of the physical control methods, and one of the chemical control methods that we're going to be talking about, are degerming methods, where you're simply removing the microorganisms. You're not killing them. Sometimes that's the best that we can do with either what we're trying to apply control methods to, or for the organism that we're talking about. So let's move on to three terms that I'm going to go over in more detail. antisepsis, disinfection, sanitization, sterilization. So, since I'm down here, let's start from the bottom. Sterilization is the destruction of all microorganisms and viruses in or on the object. Okay, so that's sterilization. We've killed or removed everything. Sanitization is the removal of objects, pathogens from objects to meet public health standards. We're going to talk about those public health standards in more detail on the next slide. Disinfection and asepsis are the reduction of numbers. Okay, so antiseptics are an antiseptic, and disinfection are not sterilization. We are not talking about complete kill. Let's take antisepsis, for example. We're talking about living tissue. If you were to try to sterilize, say, living skin, you would burn it. We don't want to do that. So it's impossible to sterilize living tissue. It's just not going to happen. We do the best we can. We lower the numbers of microorganisms and viruses so that hopefully it's to the level where the immune system can handle it. Disinfection is a similar kind of thing. For most things, it's impractical to try to sterilize, say, a tabletop. What you'd have to do to sterilize it would destroy it. So once again, we look for lowering of numbers to the point where your immune system hopefully can handle it. Now with pasteurization, we're basically doing the same thing in things that are intended to be consumed. Alright, so having gone through the official definitions of what these terms mean, let's talk about legal definitions. Sterilization, as you remember, is the complete removal or destruction of all living microorganisms on a surface. A non-living surface. Remember, we can't do that on living tissues. But how do you prove that? It's impossible to prove. So, the legal definition, if you want to get approval for a sterilization procedure, or if you want to get approval for a sterilization chemical, what you have to do is you have to show that this procedure or product produces at least a 6-log reduction of thermoresistant endospores. So what does that mean? Now remember, endospores, they're a resistant resting stage of a bacterium, and if we're talking thermoresistant endospores, these are from thermophiles. So they're going to be extra resistant to heat than your average endospore. So what are we talking about with 6-log? That's basically a 1 with 6 zeros. So you put 1 million organisms on a surface that you're going to test, you do your sterilization procedure, and if you can't find any living endospores afterwards, you're good. You're golden. So you see that we haven't proven that we have killed everything, okay, but we've met the legal standard. Now the same thing happens with antisepsis and disinfection. When you're getting procedures or products approved through the government, either the EPA or the FDA, you have to meet certain standards. Just saying that you're lowering the amounts just doesn't cut it. You have to have some sort of benchmark. So, for antisepsis and disinfection, you have to show that you can have at least or greater than a 3-log reduction of viruses and or vegetative microbes. Notice we're not talking endospores here. Antiseptics and disinfectants we do not test them against endospores. Sometimes we do if we're looking at certain things like, I don't know, trying to clean up post offices that have been infected with anthrax endospores. Then, you up things. So what's a 3-log reduction? That is a 1 with 3 zeros. So we're talking 1,000 organisms. That's not nearly as much as 1 million. Okay? Remember, we're talking about getting things to the point where your immune system, hopefully, will be able to handle it. Now, if an antiseptic or a disinfectant product, or a product that they hope to be able to pass antisepsis and disinfection, doesn't make the mark, has less than 3 log reduction of viruses or vegetative microbes, then it becomes a sanitization product. For most people and in most situations, you're not dealing with really scary microbes, so sanitization is plenty. If you're in a restaurant, in a daycare, or in your home, sanitization is just fine. If you're working in any place where you're in a clinical environment, you're seeing lots of sick people come through, then antisepsis and disinfection is important. Or if you have norovirus show up at your campus, you definitely want to go for high-level disinfection because you know you've got a particular organism that you're trying to kill. Hopefully that all makes sense. Now that we've talked about some of the terminology, let's talk about what we're trying to kill, what we're trying to control. What we're looking for with microbial death is the permanent loss of reproductive ability under ideal environmental conditions. So if they can't reproduce, they're not going to make more, they're effectively dead. So when we're calculating the microbial death rate, usually we have a constant over time of any particular set of environmental conditions. So if I want to see how well bleach kills my organism over time, I'm going to use the same temperature each time. I'm going to apply it for the same amount of time. Usually we go with 1 minute. I'm going to make sure that my bleach is the same pH, whether, clean surface, dirty surface. We're going to keep everything the same, and we're just going to look at the different microbes dying over time. Say I start out with 1 billion organisms. After 1 minute, if I have 10 to the 8th or 100 million, then I've had a 1 log reduction over that 1 minute. So that for that particular microbe, I have a 1 log reduction death rate for whatever I'm using. Now, the more susceptible organisms have died, the hardier have survived. If I apply my procedure for another minute and I get another log reduction, you can see how this is my microbial death rate, and I'm going to get a linear decrease over time. This is something to consider when we start talking about physical and chemical control methods. Some of them are time dependent. So if you're using those products or using those procedures, do not think that you can cut down on the time, because you will end up here instead of down here where you want to be. Now, the last subtopic for this topic is, how do these antimicrobial agents act on the microbes? Some of them alter cell walls and cell membranes. They punch holes in the walls, and then the cells pop in the hypotonic solution that they find as their home. If we pop holes in the membrane, they can also leak. That can lead to cell death. Now, one of the most important ions that leaks out is potassium. Cells control sodium chloride and potassium, but usually potassium is a little harder to come by than, say, sodium chloride. So they want to keep control of this. If you pop a hole in the membrane and your potassium starts leaking out, then, it's not a good thing. The second thing is damage to proteins and nucleic acids. You've heard a lot about denaturing proteins. That's because it's important. We can use procedures to denature these proteins. They're no longer effective at doing their jobs. The cell dies. Fatal mutations. If you have a fatal mutation in the nucleic acid, you can't produce the proteins that you need or the enzyme that you need, specific protein, then you die. And ribosomes. If you inhibit the action of the ribosomes, you're hitting both proteins and nucleic acids. So pretty much the action of all microbial agents are attacking either the cell walls and the membranes, or they're causing damage to the proteins and the nucleic acids inside of the cell. So here are reminders of what you need to learn for this topic.